chemistry and inorganic chemistry, the naming is different. Some people think it's easier in organic chemistry, some people think it's easy in inorganic. I think the organic is pretty easy in here. When we go to name things, the prefix is going to be based on the number of carbons in there. So if there's 1, it's math 2F 3 pro 4 U. So you just look at it and go, okay, this is how many carbons, this is what you use. These are also functional groups that get put into names things. Then you look and go, does it have a single bond in it? If it does, it has an ANE, so methane, a single bond. If it's got double bonds, it's going to have the ENE, so ethene. If it's got triple bonds in there, it's going to have a YNE in there, so butane in there would be an example. So it's pretty easy to use based off of that. So you can just look and go, how many bonds are in there, how many carbons are in there. And that's how you do the naming for these smaller ones. Does that make more sense to you?
What is the AL and ethanol?
That's an alcohol ethene. So if you have that down here where you had that table of groups, ethanol, it's the OH in there. So they make sense. You're just getting the ones for the forced, for the simple naming like that, and that's good enough because I'm not going to ask you to name them. It's just so when you hear the names, you go, oh yeah, there's some method behind the madness in there, and you don't sit and go, I have no idea what it is because the name is so weird to you. Did I give you what you needed to know out of this section, out of chapter 4? Okay. So how is the stuff with the isomers? Did that make sense? Do we have any questions on that? I know that was
over it.
Okay. So an isomer just means you have a variation in the architecture of the organic molecules. So if you've got 4 carbons, or in this example here, it gives us 5 carbons, these are structural isomers. You just rearrange the 5 carbons differently. So here it sticks out to the side instead of being in a straight line in there. And they will typically have different properties if you arrange them differently in there, because so many things work on along the key mechanism. A cis and trans isomer is specific when you have a double bond and you've got 2 different substances in here. So if they're next to each other, we call them cis, like sisters, is the way I remember it, next to each other. Trans, they're diagonals, like that. So can you think of any examples where you might run into a cis or trans isomer? Trans fats. So that is where we see that in common everyday stuff. Normally when the body has fats, and what occurs in nature are a nice mix of these in there. They do have different properties in the body. So when they artificially hydrogenate things, and the reason they do it is it gives it a longer shelf life in there. It's not that it makes it better for you, just longer shelf life, cheap to do. It will make a whole lot more of these trans isomers in there that are going to be made differently in the body. And so while they originally thought, okay, you know, if we artificially hydrogenate these things like this, we can make the fat seed more solid. So we'll make this lovely stuff margarine or Crisco. So if you've gotten butter on your hands, and then maybe margarine or Crisco on your hands, Crisco is the worst. You cannot get that stuff off or anything. Well, these have different enzymes that break them down. So when they're in your body, if you eat a whole bunch of this, your body doesn't get the right thing in there. So we know that this is going to be associated with more flaccid. So while they did this to try and help give people an alternative, because the saturated fats and things like butter and lard, they taste good, so we tend to overdo it in there. Instead of maybe getting humans to realize you're overdoing it with this, this is where the problem is. Let's let them keep overdoing it and we'll make something else that fits in the place. Well, what they made to fit in the place is actually worse than the butter and lard for heart disease in there and increases more plaque. So, same side, opposite sides, acts differently chemically than the other one. And then the other type of isomer is the enantiomer. And these are mirror images. We kind of think of them as being handedness, right and left-handed versions. So with this, what you have to have is around a carbon 4 different things around it. And it has to do with the way that it spins light, which I have no idea what was going on in somebody's head when they decided they wanted to see if molecules could spin light. You know, I think like a biologist, not a chemist, so I just accept there was a reason they did that. But they're in a different order, and so what it's going to do is move light differently. So like a right-handed version versus a left-handed version. And these are also going to behave differently in the body. One example of it is that drug thalidomide in there that was used for morning sickness years ago. And it would go into the body as the good enantiomer, and then the patient's body would convert some of it to the bad form in there in the liver. And there's no avoiding getting the drug in your liver, so that's not something that they could get around in there. But what it would cause is to have birth defects in there. And so if they were taking it for morning sickness, something that causes birth defects is not a good plan. So thalidomide was pulled off the market eventually in there. Thalidomide is back on the market for limited use. It is not used for morning sickness because it did actually have some other good effects in treating certain cancers in there. But it is mandatory that a person be on birth control, have their tubes tied, or somebody that they're with has a vasectomy, and they agree they are going to be messing around with anybody else in there because of it's such a high incidence of getting birth defects with it. They don't want to take any chances of somebody getting pregnant while they are on it. So does that make sense? It's a right-handed and a left-handedness in there. You're not going to need to look at something and decide is it right-handed or left-handed in there. Just know that it can occur either way.
What do you mean by move the light? Or
they would put light through it, and it would the way that it rotates or bends the light in there. Yeah, similar to a prism in there. There's equipment that they will use that you can put light spectrums through there, and it's going to reflect different spectrums, and it's ways of helping to identify chemicals in there. And I suspect that may have been what was going on when they used this because, like that knowledge with thalidomide in there, we didn't understand why it caused birth defects for quite a while. So is there any other questions in here? All right.
As for the isomers though, is it just going to be definition? And then
Yeah.
Okay, so the picture.
Okay, so with Unit 5, now we're looking at the large biological molecules or macromolecules. These are going to fall into 4 main classes: your carbohydrates, lipids, proteins, and nucleic acids. The nucleic acids are what your genetic material is made of. So these are going to get into some things that are a little bit bigger that you actually think about being in cells. You think about eating carbohydrates, lipids, and proteins in there. Most of them are going to be polymers, which means it's going to be a long molecule that's going to be made up of many similar blocks. I forgot to put the leash on to start recording. So there's going to be a blank spot at the beginning. Just fast forward through it. So with the polymers in there, oh, a stick. Now I really feel like a dog chasing a stick here. So with a polymer, you've got all these little pieces that are kind of like Legos in there. And you just hook each of those individual pieces together to make a chain. Or kind of like those pop beads that maybe you had when you were really little, way back when. So that you can make the chains with them. So the monomers are the individual units, and it's a polymer when we hook multiple of them together. So when we go to make these, most of the time it's going to occur through a condensation reaction in there. And when we look at these synthesis reactions, there's a couple ways in here. The dehydration is also a condensation reaction in there. When you dehydrate something or it condenses, what's going to happen to it? What are you doing with water? So when you dehydrate something, you're taking the water out of it. And that's what you're doing here. You can see on the ends here where you've got this hydrogen, this hydroxide, they're going to both get pulled off and made into water, and in turn for removing these off of there, it allows these two pieces to come together. Oh. So hydrolysis is going to be the opposite in there. Hydro means water, lysis is to split. So you're putting water into it so that it can fill in that space there and split it apart. And for me, the easiest way to remember that is to just really think about the name. Let the name give you the hint. Lysis, to split, is what that means in there. And then dehydration, when you dehydrate things, you pull the water out. So one of the things we can use these macromolecules for is as enzymes to help speed up chemical reactions in there. So chemical reactions are going to go at different rates depending on the environment around them. And an enzyme is a way that you can help to speed that up when you can't change the environment that's around you. So the first group we're going to look at are the carbohydrates. These are your sugars and the polymers of sugars. So mono means one, saccharide means sugar. So monosaccharide is one sugar, disaccharide is two, polysaccharide is many. And we can classify our sugars different ways. So they can fall into the category of being aldehyde or aldoses or ketoses or ketone sugars in there. An aldehyde sugar is going to have this double bond with oxygen on the end. A ketone sugar is going to have it somewhere in the middle in there. Sugar. Yeah, it's just another name for sugar. So then we look at how many carbons they have. So a triose has 3 carbons in it. So the example it gives is glyceraldehyde and dihydroxyacetone. So the dihydroxyacetone, anybody have any idea where you might find that sugar out in the world? Dihydroxyacetone. So ketones stink, right? If anybody's ever used self-tanner, it's dihydroxyacetone in there, and that is why you stink when you put it on. If you didn't know that you stink, you do when you wear this stuff. So it has a very characteristic smell. They will try and perfume it, but ketones just have a distinct smell to them. So pentoses, penta means 5. These are 5 carbon sugars, ribose and ribulose. And then we've got the 6 carbon sugars. These are the ones that you'll run into the most. Ribose is in the RNA, but in DNA, but we still see these are what we think of when we think of sugars in terms of food. So glucose, galactose, and fructose in there. Those are the ones that taste good. And they're actually in other carbohydrates too. It's just they're in longer chains than the other carbohydrates. So they're checking for just glucose in the bloodstream because your body is going to, other things that come in, you've got this way that we'll talk about later on in the quarter of breaking down glucose, this chain of things. And if you eat something else, it's going to get converted in and join that main chain. So monosaccharides are going to be the single sugars in there. All sugars are multiples of a carbon, 2 hydrogens, and oxygen. Glucose is the most common in there. So a disaccharide is going to be 2 sugars in there, and they're joined by a glycoside linkage, which isn't going to mean much to you right now. Maltose is made up from 2 glucoses, lactose is made up of glucose and galactose, and sucrose is made up of fructose and glucose. So maltose, this is in starches. Where would you find lactose? Milk. Sucrose. That's table sugar. Yeah, the table sugar that you put on your table. So polysaccharides are going to be multiple of these put together in there. And so starch is one example. It's a glucose polymer that you find in plants that have plastids, including the chloroplasts. So any idea of some plants that would be high in starch? Potatoes. Usually if it's got a root that grows in the ground and we eat the root, that's usually going to have a fair amount of starch in it. What's up above the ground will not typically be starch. No. The onion's just really superficial in the ground. It doesn't have like the tuber root. It's a different kind of structure in there. So the potatoes are a good example of that in there. Glycogen is the polymer of glucose that gets stored in animals. So that's going to be what you store in your muscle and liver for sugar in there. And different starches are going to be glued together in different ways. So when we say this 1,4 linkage in there, it means carbon 1 is attached to carbon 4 in there. And if you hook them together differently, it takes different enzymes to break it down in there. So amylose is a simple starch. It's unbranched. Amylopectin, it's a little bit different. It's got 1/6 linkages in there. So any ideas where you would find amylopectin? Yeah, the fruit pectin that you use in jelly. Cellulose, this is another one, and it's got different bonds yet in there. It's going to have the alpha and beta in there. Cellulose is all beta. It's straight and unbranched, but there's very few organisms that can hydrolyze the beta linkages of cellulose. So can you think of anything you eat that's got cellulose in it? Corn. So what happens to the corn when you eat it? Nothing. The inside of the corn you can digest, but the outside of the corn that's made up of cellulose comes out the same way it went down when you chewed it. So if you didn't chew it very well, it's going to be a pretty good indicator. You could look in there. If you wanted to actually pull those out, you would see it's the little coatings on the corn that you did not chew up all the way in there. Grass also has a lot of cellulose in there. So if you have a dog, dogs tend to like to go snack on grass, and for some reason they like to pick the longest blade of grass they can find. So what happens when the dog eats that grass? Sometimes they vomit, sometimes it goes through, and it's not digested in there either. So you may have the long piece of grass that's hanging down from the back of the dog in there. So when you see them eating the long grass, steer them towards the short stuff. You know, one morning I got up and I found what looked like he had taken an ice cream scoop of short grass. He'd gotten into the grass clippings. And I look and it's like, who took an ice cream scoop full of grass clippings and put it on my carpet? Better that end than the other end, though. So chitin is another polysaccharide. This is what's in arthropod skeletons and a lot of fungi. So we can digest this one in most cases in there, most forms of chitin in there. So if you're wondering, can you eat bugs? Yes. In there, most bugs you can eat. In our culture, we tend to look at it and say, oh, that's just gross. We wouldn't do that. But there are plenty of cultures out there that they are considered a delicacy. I've heard some of them are kind of good. I've never tried it. Shrimp in there, you can actually eat the outside of the shrimp. So there are some Asian restaurants that they have like deep fried shrimp heads. I was feeling adventurous one night, so I tried it. It wasn't bad. It tasted like anything else deep fried. If you know, kind of was a reality check to me that if you deep fry the bugs, I might not know the difference and I'd eat it. It just tasted like anything else deep fried. It was just sharp, and that was a little disturbing to me. The thing kept poking inside my lip, And I'm like, I'm not looking for a piercing from this. It was just the head.
Just the head, no body.
Right.
I'm thinking it's like a whole.
No, it was small. It was bite size. And I tried all kinds of weird things there that night, so the shrimp head was the better tasting of them. So this picture here just shows how you've got the differences in these linkages in here. You'll notice these have them all pointing down. These, they alternate going up, and down in there. So the difference between, you can digest it, you pull it out the back end of the dog. So your lipids, these are all going to be hydrophobic. It's going to include waxes. Some of your pigments are lipids, fats, phospholipids, and sterols. Fats are going to be constructed of a glycerol and fatty acids. Most of the time in humans, they're going to be in the form of a triglyceride, where you'll have 3 fatty acids attached to a glycerol. A fatty acid has a long carbon chain. It's usually 16 or 18 carbons. In humans, in there at the end, you have a carboxyl group. It's relatively nonpolar, so it means it's not going to like to mix with water. Glycerol is a sugar alcohol in there that's got 3 carbons, and each of them is going to have an alcohol group on there, that OH group. So when we look at fats, we look at, are they saturated or unsaturated? Saturated means that it's attached to the maximum number of hydrogens in there. All of the bonds are going to have hydrogens attached to them instead of sharing with a neighboring carbon twice in there. Where an unsaturated fat is going to have 1 or more double bonds in there by removing one of the hydrogens. And what happens when they're unsaturated is it puts a little kink in the molecule. It changes the shape. And my little picture of it's down here. So with your saturated fats in here, it's a nice straight molecule. Unsaturated, there's a bend in it. So when you go to stack them up, if you have a nice new ream of paper and you set that on the counter, that's going to be a lot more stable than if you have papers that have been all wadded up and bent and you try and stack those in a pile on a countertop in there. So what you'll end up with, the saturated fats, more stable, more solid at room temperature, unsaturated, less stable, liquid at room temperature. So which one do you think would be more stable to cook with then? The saturated fats in there. And that's why you'll see a lot of places advertising that they use like peanut oil or coconut oil to cook things, is the nut oils are more saturated than the vegetable oils or canola oils in there. So what happens with these when you cook them is they'll get more free radical damage in them in there, more likely to go rancid. So if you've ever had like some oil that maybe you found in the back of your pantry and you go, oh, I forgot I had this and this has been in there a while. And you smell it and you go, oh, it smells weird. And you go, well, I'm not going to throw it away. I bought it anyway. And you cook with it and you go, oh, tastes weird in there. That's the rancid. Yeah, and you just gave yourself a whole bunch of free radicals. With what oil? Yeah. Yeah, I mean with the, most of the time if somebody does deep fry things, they do save the oil because you have to use so much of it, but you don't want to save it for months on end in there. You know, even fast food restaurants change that stuff weekly. Yeah, if they're disposing of it after a week, that should tell you that, oh, that better go no more than a week. So the trans fats, this is that process of hydrogenation in there that we talked about earlier. The phospholipid, this is 2 fatty acids that are attached to a glycerol, and instead of having a fatty acid at the 3rd spot, what it's going to have is a phosphate group in there. And that's going to create an area that's polar in there. It's going to have a slight charge to it, which means it's going to like to interact with water. And there should be a picture of this in here. So this represents your phospholipid. They're typically drawn like this to have the little head on them with two fatty acid tails. What this represents is your phosphate head in there. It's hydrophilic. It likes water. Hydrophobic. This end does not like water. So in cell membranes where you have this, the way we will arrange those is you have two layers that are in opposite orientation in there. So this way you can have watery substances inside the cell, watery substances outside the cell, but the watery substances would have to cross through here to mix, and they don't want to do that. And then we have the sterols, and these are going to be lipids that have these 4 fused rings in them of carbon in there, and then they'll have different functional groups attached to them. Cholesterol is the most common one in there. It's found in animal cells. This is the precursor to making other steroid hormones. They're called steroids because they're derived from sterols in there. And cholesterol may contribute to atherosclerosis with the trans and saturated fats in there. And I guess I did not put a picture of a cholesterol molecule in there. You'll get to see it later on. Then we have the proteins, and this is what structures are made up of. This is over 50% of your dry mass of most of your cells in there. The only exception would probably be your fat cells in there, and they are mostly fat. So types of enzymes, they can have enzymatic actions in the body, so they'll act as enzymes to allow reactions to occur. Defense proteins in there, so these are going to be what makes up part of your immune system. Your antibodies that are going to attach to invaders that come in are proteins. You have storage proteins that can store amino acids in there. So an example would be the ovalbumin. When you look at eggs in there, you've got the egg yolk, and then you've got the egg white that's going to have a lot of that ovalbumin in there. The purpose of the egg white is to feed that egg yolk when it turns into a baby chicken. Transport proteins that are going to move things across membranes, ones that will act as hormones, some that will act as receptor proteins. Some are going to be contractile, so they'll actually be in your muscles to allow them to contract and pull, and then other structural ones that make up things like collagen. So proteins are going to be polymers as well, and they're all going to be made from the same 20 amino acids. So when we look at an amino acid, they will all have a similar structure. We've got a carbon, a hydrogen group on one side, an acid group on one side, an amine group. So those are showing them as their ion forms with this hydrogen coming off onto here for a binding. And then they have this R group, and that R is what's variable. This is the only thing that's different in all 20 of them. Everything else is the same. So this shows pictures of them here where you've got R and, well, they didn't just highlight R, they highlighted the amine and the acid group in there. So we can classify them based on some of their properties in there. So some of them are polar, some of them are non-polar in there. Some are going to be electrically charged and be acidic. Some are electrically charged and are basic. Some are considered essential, meaning we have to consume them in our diet. We can't make them out of other things. And then some are non-essential. These are ones that we can make out of other things if we don't eat them. Where that usually comes into play, if you eat animal products, you're going to be eating all of the essential amino acids. But if you are a vegetarian or a vegan and you don't eat any animal products at all, vegetable products, there is very few that actually have all
of the essential amino acids in there. So it's just important to have more variety in the diet if a person is a vegan. If they eat the same thing all the time, they eat just, you know, the same few plants, there may be a few of these that get left out in the mix in there. And they used to look at it and say, oh, you had to do this food combining and it had to be eaten in certain meals. And there was vegetarians for years that never did that were still alive. And now they've changed their thoughts on that because all those people weren't dead. And realized as long as you get a variety in there. So sometimes people will look and go, oh, well, they're a vegetarian, they're going to be healthy. Not necessarily. There are lots of junk food options you can get that are vegetarian out there. So the branched chain amino acids, the BCAA, these are ones that are metabolized in the muscle rather than the liver. Anybody that you can think of that might have a particular interest in consuming more branched chain amino acids? Weightlifters. So if you're around bodybuilders at all, when you look, you watch them go into a supplement place, you're going to hear them talking about getting their branched chain amino acids while they're looking at their whey protein and eating that with their chicken and rice in there. I hung around with bodybuilders when I was in school a lot, and I know a lot of their little idiosyncrasies in there on that. And it was just their little protein drinks and stuff, they weren't going to budge on that stuff no matter how much you teased them about it. So your amino acid polymers are going to be linked by a peptide bond. And the peptide bond is just a covalent bond that's formed when you have this group next to this group in there. So what you're going to do is you're going to pull off an OH here and a hydrogen here, and you'll have dehydration that occurs, and then they bond together by removing the water out of it. So proteins fold up to have your three-dimensional architecture. So where we looked at that picture of morphine last time, going in and how it fit into that endorphin receptor, all of the proteins fold up to make three-dimensional things, and that's how they will interact, is they have to fit into another three-dimensional slot that is meant specifically for them. Some of them will be more globular shaped, some of them will be more fibrous shaped in there. But they all have what we call 4 levels of protein structure. So your first level, or your primary level of structure, is the sequence of amino acids in there. So what order are those 20 amino acids, or the different amino acids in, and which of those 20 are they? So it may be a lot of leucine, isoleucine, valine in there, made up of certain things. That order matters. If you change the order in there, it's going to fold up differently and you're going to get something different made. The secondary structure is going to be where it's going to either fold up or coil. So it can make alpha helices or beta pleated sheets in there. You can actually have different regions of it turn into beta pleated sheets and alpha helices within the same protein in there. So when you have those beta pleated sheets in there and these alpha helices, you get other parts of it that are closer together, and then you have hydrogen bonds in there that help hold them in that shape. The tertiary structure is going to be the overall shape. So after you do those coils and folding, you fold it up some more. So here you can have regions where you have hydrophobic interactions, where you can make little clusters of it that are going to be out of contact with water. Some of them have sulfur in them, and when you have more of those get close to each other, like 2 cysteines, you'll have these sulfhydryl bonds that will occur in there, where you have 2 sulfurs that bond together. Those are going to be important because those are really strong bonds in there. So places where you would have a lot more sulfur content in there are places like your hair and fingernails. It's what makes them hard. So your quaternary structure is the overall structure when you have multiple polypeptide units come together. So you've made one here. Here the quaternary, you got 2 of them that come together in there. So this here is kind of to give you an example of how one small little change in there can make things go really wrong. So here's your primary structure, your normal sequence of hemoglobin. This is a sequence of hemoglobin here where just one of the amino acids changed. So hemoglobin normally has its secondary structure like this. Here it's a little bit different. It folds up, goes on to make the quaternary structure, and it's still going to be different in there. What it results in is the hemoglobin making the red blood cell sickle-shaped rather than this round shape in there. So it only takes the mutation in one letter to create sickle cell anemia in there. So sickle cell anemia is actually fairly abundant in the population, particularly in anyone of African descent, because in places where there's malaria, somebody with sickle cell trait or sickle cell anemia is going to be more resistant to having severe malaria infection in there. So that ended up surviving in the population because a lot of times you would have lots of little kids die of malaria that never went on to breed, that were maybe normal red blood cells in there. So you just had fewer normal hemoglobins in the population over time. So when proteins denature, they lose their shape and become biologically inactive. Sometimes in the lab this can be reversed, but a lot of times when something denatures in a natural setting, it's not going to be reversible. So an example would be an egg. If you heat it up, you're going to denature it. Its properties are different. You might be able to use a raw egg to make some cookies. If you put a cooked egg in your flour and stuff like that and try and make cookies, it's not going to work. Different properties altogether. So an example where this would affect humans is when they talk about watching the fever and not to let it get above 105. That's the temperature where a lot of your proteins begin to denature in there. And that's why they don't want a fever to get above 105 in there, is you can have permanent damage if the body temperature gets above that. So your chaperones, these are chaperone proteins in there, and they assist with the proper folding. So what they do is they are going to help make sure things stay folded properly. So how do we know these three-dimensional structures in there? They can use X-ray crystallography in there. So they use an X-ray and diffract the beams on there to be able to look at the shape of it. They can use nuclear magnetic resonance spectroscopy in there. You don't have to crystallize things to look at it there. And then bioinformatics. We've got the shape of enough proteins now that a lot of times using the amino acid sequence and knowing how it's going to behave in there, they can predict it just using a computerized model in there. So the next section, nucleic acids, this is your genetic information. And some of this information you've had before in there. The genes, that's going to be the unit of inheritance. It's going to be what determines that sequence of the polypeptide. It's the instructions for making that polypeptide in there. The nerd that makes the instructions. So it comes in two forms, DNA and RNA. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. You'll almost always just see those as DNA and RNA. I think they're in the dictionary as DNA and RNA without the full word behind them now. The chromosome is going to be the form that you have during cell division in there when you're going to replicate things. So you've got the DNA and it's normally all spread out. The chromosome is when you wind it up and coil it into nice little chromosomes in there so that it's nice and organized when the cell divides. So typically a chromosome will have over 100 genes. Right now they think the Y chromosome has 78. It's the only small one. Your mRNA is your messenger RNA. When you go to make proteins, you don't want to send out your only copy of the gene into the cytoplasm of the cell to the workers to say, here you go, can you make a protein out of this for me? If that's your only copy of it and it gets destroyed out there, you're not much good as a cell anymore. So the mRNA is like a photocopy of it almost. It's a copy that's made that gets sent out there so that the ribosomes can actually make that. So the ribosome is your organelle of protein synthesis. It's slightly different in there. It uses uracil instead of thymine. So it's slightly smaller. You can just stick with the definitions on here. But you have to use it because you can't fit a DNA through the pores in the nucleus. You can fit an RNA through. So a polynucleotide, that's just a polymer of nucleic acid macromolecules in there. When we have a nucleotide, it's got 3 parts to it. It's got a nitrogenous base, a 5-carbon sugar, which is the pentose sugar, and a phosphate. A nucleoside is only 2 of the parts, the pentose and nitrogenous base without the phosphate. Not really sure why they decided to distinguish this and name them 2 different things in there, but you will see both names used. The way I remember it is you have 2 things before you have 3, you have S before you have T. It's one of those things that most of the time when you're looking at something and you see the term nucleotide or nucleoside, it's not going to be critical that you remember that it's got 2 or 3. You're thinking it's the part of the DNA structure, and you just move on from there. Yeah. So that's how I would memorize them for saying which one is which. But most of the time when you look at things, if you forget that, it's not going to be a make it or break it deal for you. So your nitrogenous bases in here, they fit into 2 categories, pyrimidines and purines. Pyrimidines are 6-membered rings in there, and they're going to include cytosine, thymine, and uracil, the C, T, and U. The purines are a little bit larger. They're a 6-membered ring fused to a 5-membered ring, and they're going to include A and G, which is adenine and guanine. The way I remember it is you've got purine here, so pure as gold. That's the way I remember it in there. Because otherwise they are just names. Yeah. And everything else is a pyrimidine. When you have it bound, you're going to have a purine and a pyrimidine at every level in there. So ribose, just the 5-carbon sugar in RNA. Deoxyribose, the one in DNA. It's just missing an oxygen in there.
So how do you memorize the C, T, and U?
That they're pyrimidines, they're the leftovers.
Oh, okay.
So when you look at these carbons in there, you've got each of the carbons is numbered in there so we can distinguish where things are on there. And we've created this arbitrary reference point on there for the 2 ends of the DNA, that the 5' is going to have a phosphate attached to carbon number 5. The 3' end is going to have the hydroxyl attached to carbon number 3. So when you look at the string of DNA, one has carbon number 5 open and free on the end. The other end of it has carbon number 3 open on the end and there. Not of the things on there, but you have it so that it's oriented. You've got 5' at one end, 3' at the other. And there. And the reason we need to know what end is what is it's going to build 5' to 3'. So it adds on to this end here. The enzymes only read it in one direction. It goes 5' to 3'. Always. So there's also purpose in that you would want to make certain proteins or read certain genes before certain other ones in there. It would be kind of equivalent to saying I'm going to make saliva before I make a mouth for it to go in. It doesn't make sense. And then they're arranged anti-parallel. So instead of them being lined up like this, they're lined up like this. 5' to 3' one way, 5' to 3' the other way. Always. It just is. It's the way they fit. I didn't design the body. There would have been a few things I would have done differently if it were up to me. So with the DNA, it's a double helix in there. You've got this model in there, the two sides of a ladder that twist up in there, in the anti-parallel arrangement. So the way things are going to pair in DNA, you're always going to have A pair with T and G pair with C. So they fit together here. You can see that they're complementary shapes in there that they have to pair together. You can't put the pokey end with the round end. In RNA, A is going to pair with U. So you have uracil instead of thymine in RNA. Adenine, A. And rarely would you ever have to write out the full name of a base pair. We always just abbreviate them using A, U, G, C, and T. This is just kind of an overview of what happens when this process, when you go to make a protein. You've got the DNA that you use to make this mRNA photocopy. That fits through the pore in the nucleus, goes out to the ribosome, who reads it and makes these polypeptides out of your amino acids. That's just an overview of what you'll see in there. You're going to get it broken down into more detail more times than you even want to think about. The nucleus is in the middle, and then out here is the cytoplasm. So with the DNA in there, when you did that one thing in the evolution worksheets in there, it had you comparing these sequences of amino acids in different species in there. What you find is siblings have greater similarities in DNA than other unrelated individuals of the same species. So your DNA is more like your brothers or sisters than for somebody else. So when you're looking like for organ transplants and things like that, where they've got to look at some of the markers on cells, that's why they look to your siblings first. Doesn't mean there wouldn't be a random stranger out there that would have something similar, but the trick is finding them in there. So this is a more detailed picture that shows that 5' end, the 3' end in there. I would not beat myself up and say try to learn to draw this or memorize this. That's just kind of insane. But you can see there is reason behind this 5' and 3', and if that helps you, just put that in your mind and say, oh yeah, there is reason behind that, and I've seen it, so you can let that go. That's what I would do with it. Yeah, there are some things that with the chemistry, as you're going through, you get to see it enough that you accept it and let it go so that you can focus on what you really want to learn. So that's the end of that section here. Why don't we go ahead and take about 5 minutes?